This is the process of transcription. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. During transcription, the DNA in the gene is used to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of the gene expression is controlled by blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. During elongation, the RNA polymerase slides along the template DNA strand from the 3' to 5' end, generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end. Unlike replication, RNA polymerase zips DNA back up, keeping only 10 to 15 bases exposed at a time. Once the RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene, and the DNA is returned to its original state. The strand of messenger RNA that is made during transcription includes regions called exons, which code for a protein, and introns. The only way the mRNA can be used in translation is by removing the non-coding introns. Also, a 5' cap and a 3' poly A tail must be added to the mRNA. This process is called intron splicing and is performed by a complex called a spliceosome. Spliceosomes remove the non-coding introns. Introns are cleaved at the 5' GU sequence, which is the starting code for introns, and forms a lariat, or loop, at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, which is the end code of an intron. Once all of the introns are spliced out, the exons are linked together to produce a mature mRNA strand that can then be used in translation. The mRNA strand that was produced moves from the nucleus to the cytoplasm, where it will find the ribosome. This is where translation occurs.
In molecular biology and genetics, translation is the process in which ribosomes in a cell cytoplasm creates proteins. Translation occurs inside a ribosome. The small ribosome subunit binds to an mRNA and an initiator tRNA, which begins the sequence. First, the mRNA acts as a code for each specific protein. The protein is determined by each of the 3 bases on the mRNA, known as codons. The mRNA sequence, determined based upon the codons, then gets matched with the anticodon, which is then carried by the tRNA, also known as transfer RNA. The large ribosomal subunit joins the tRNA and mRNA sequences, which then completes the initiation of translation and forms the amino acids. These amino acids are covalently linked with the tRNA sequence. The process begins with the start codon AUG and continues all the way down the mRNA, forming a long chain known as a polypeptide, while removing the previous tRNA. This process continues until a stop codon reaches the end of the mRNA sequence. The 3 stop codons are UAA, UAG, and UGA, which are used to terminate translation. Once translation of a sequence has been stopped, the polypeptide will leave the ribosome and move on to other cell organelles for further modification.
Go.
Initiation. Elongation. Termination.
I can't do this. I can't do this.
Just go. I'm not stopping the video.
Okay. Oh, are you going?
Mm-hmm.
Oh, okay.
I wanted to make sure I started.
Transcription.
No.
Ready? 3, 2, 1. Do it!
Termination occurs. The enzyme detaches from the gene. Oh, that's when you're supposed to do that.
Stop!